It's a pleasure to introduce the next speaker, Professor Viola Vogel from the Department of Health Sciences and Technology Laboratory of Applied Mechanobiology at ETH in Zurich. And the topic of her talk will be bacterial nanoglues targeting wound sites for theranostic applications to read out tissue fiber tensions in health and disease.
So thank you very, very much, Andre, for the introduction. I have some fundamental questions to you. Beyond all our challenges that we have in drug delivery, are there other challenges where we know rather little about disease progression, cancer, fibrotic disorders, and where our nanotech tools can make a huge difference in understanding these challenges? And so what I would like to show you here is the very first tissue cross-section ever that stands for the tension of tissue fibers. So far, we had not a single tool that allowed us to measure the tension of tissue fibers. So as I go through my talk, I first would like you to appreciate why having such a tool available is important, but then even further, how this tool works and how we discovered it. So many ECM transformations have been reported to drive cancer progression. But exactly how this works is not known. So we know that the matrix gets stiffer, but how that impacts cells and how the cells that reside in the tissue contribute to this phenomena is not really known. The same is true for ECM transformations that drive the progressive fibrotic disorders. Again, exactly how it works and under what conditions TGF-beta released is again not known. So why? So the nanotech community has made huge contributions in the last 20 years to stretching proteins and seeing that protein structure functions can change. They made major contributions to designing and fabricating devices that can measure cell tension and forces, and to a number of other nanosensors, so that we learned enormously about molecular biomechanics and cellular mechanotransduction processes. And that field is today called mechanobiology. But then we have all these observations at the tissue level, but we don't know how to connect across length scales. We don't know how to connect the knowledge that we gained at the cellular level to the tissue level. So since all the nanoprobes we and the community developed previously, many of them based on FRET, are very hard to apply at the tissue level, we asked the following question. Can peptide probes be developed that measure the mechanical strain of extracellular matrix fibers at the tissue level? There was one attempt by Tom Barker who screened random peptide libraries. He found a few peptides that bound rather weakly to stretched versus relaxed fibronectin fibers. So we asked the question, can we take advantage of nature's evolutionary process? The protein that we are particularly interested in is fibronectin. It's always upregulated in wound sites. It's upregulated in cancer. It's upregulated in fibrotic disorders. So what is very interesting is bacteria evolved many adhesins that bind to fibronectin. And the first forms of fibronectin are reported already in fish. So there's a very long evolutionary process by which bacterial adhesins and fibronectin co-evolved. So now I pose a really interesting question. Imagine you are a tiny bacterium, and you try to find the site of injury in your host. How do you do that? How do you see where you might enter your host? So it's this challenge here. How do you find it? So the extracellular matrix, if you have a wound site or a site of inflammation, the tissue fibers are always cut in those locations, mechanically or enzymatically. And this is what we found. Bacterial adhesins from S. aureus bind to relaxed fibronectin fibers much, much better than to stretched extracellular fibers. And this probably allows bacteria to bind to specifically wound sites and sites of inflammation. And the adhesins that target the N-terminus of fibronectin are not unique to S. aureus. We found 36 bacterial adhesins that work the same way. So what is interesting is the adhesins all do not bind into a binding pocket, but they bind in sequence to a few of these nodular modules of fibronectin. So now in every healthy tissue, cells do not just sit. They need to explore whether there's injury. So they are constantly pulling on extracellular matrix fibers. But as they pull, and here I show you now what happens to fibronectin, the N-terminus, if we pull, the domains get stretched apart from each other. And the distance changes. And why is this so important? The NMR complex was known for the bacterial adhesins binding to fibronectin. And here you see, you pull on fibronectin and the bacterial peptide pops off. So you introduce a mismatch in a multivalent binding motif. So it's like building with Lego blocks. All Lego block knobs can bind to a base plate, strong adhesion. If you stretch the base plate, only one knob would be able to bind. Exactly the same mechanism is exploited by bacteria to recognize relaxed fibers. So in the next step, we asked, okay, can we take advantage of this knowledge now to make a sensor that recognizes the tensional state of tissue fibers? And so we labeled the peptides, and we made scrambled peptides, and we labeled them with fluorophore and with radionucleotides. But first, let's see whether this idea works in cell culture. So many years back, we already developed a FRET sensor, fibronectin FRET sensor. We can add it to the cell medium. It gets incorporated into the matrix. And here you see a lot of FRET, and those are relaxed fibers compared to much more stretched fibers. Then we labeled the peptide with a third color. And what you see is high intensities are found in the regions where fibronectin is a more relaxed state, and we see a beautiful correlation over a large range of strains. So the idea works. So let me show you again my starting image, this cross-section. Here we stain with antibodies for fibronectin, as expected, highly upregulated. We stain with a peptide. You see some regions stain really highly for the peptide and others not. We know that tumor tissue is very heterogeneous, but nobody expected that there are areas with a lot of relaxed fibronectin fibers. And so since nothing is known at the tissue level, we start exploring in the moment, what does it even mean physiologically? So you see another lung carcinoma model, again, highly stained really, really highly for this peptide. So how can we now learn whether or not this finding is significant? So we first of all asked, do we see stretched or relaxed fibronectin fibers close to blood vessels? And here you see a lot of blood vessels in blue, but we do not see a correlation that the relaxed fibers are round or far apart from blood vessels. We saw a good correlation between thick collagen bundles that can be seen in second harmonic generation. And the correlation is so high that 87% of the fibronectin binding peptides are in close proximity to collagen bundles as shown by second harmonic generation. But then, and here comes the real big surprise to us, we also started staining for myofibroblast with alpha-smooth muscle actin. And we find a huge correlation of the presence of relaxed fibers in the proximity of myofibroblasts. And that is against everything you find in the textbook. Because we always thought myofibroblasts are the contractile cells, so they should stretch all the fibers that surround them. No, this is not the case. They produce a lot of extracellular matrix proteins and these thick collagen bundles, and collagen bundles are much more rigid than fibronectin. So the cells start pulling on collagen, and fibronectin in the proximity relaxes. Then we asked, okay, how about healthy organs? And you probably appreciate already, we see a little relaxed fibronectin, but far, far less than in all tumor tissues. And why do we really care about that? And does it make a difference? And so first of all, you need to know a little more about fibronectin. It's a huge protein. It has domains with binding sites for cells, for bacteria, for growth factors. It has many MMP cleavage sites, and not even all binding sites are shown here. So our working hypothesis over the last few years was that proteins can act as mechanochemical switches, and if you stretch proteins, you might activate some binding sites or turn others off. And in the meantime, we have a long list of how these switch, what might be switching. So we found that certain monoclonal antibody binding sites can be destroyed by stretching fibers. So if you do immunostaining and your antibody doesn't bind, it could be that you simply destroyed the epitope. The protein is there, but you don't see it. We also showed recently that fiber stretching destroys the collagen 1 binding sites on fibronectin. And in cell culture, cells can only assemble collagen 1 in the presence of fibronectin. So fibronectin acts as a template, but its templating functions is destroyed if you stretch the fibers. It has huge implications for the hierarchical organization of matrix. We also now found, and this is important for lymph nodes, that the cytokine binding of IL-7 is upregulated if you stretch fibronectin fibers. And then and this is again important in the context of cancer, osteogenic stem cell differentiation is enhanced on stretched fibers compared to seeding the cells on relaxed fibers. And this process is EGFR regulated. So if we block EGFR signaling, then the cells cannot distinguish between stretched and relaxed fibers. So what we try to establish is a model where the cells do not just read rigidity or stiffness or softness, but where the cells, as they pull on fibers, stretch the fibers, change the chemistry of those fibers, and that has a huge range of diverse implications, and that all feeds back and controls cell fate. And unless we have probes to measure this at the tissue level, we can't make progress in understanding not only this cycle, but this evenly important cycle. So you are all interested in animal studies. And so I would like to show you one proof of concept study towards theranostics. Can the peptide be utilized to target fibronectin in living animals, particularly cancer? So we radiolabeled the peptide. We injected it into the tail of a mouse. And here you see in this tumor model, the peptide accumulate in these regions in a living animal. And the SPECT-CT image was taken 4 days after injection. So this peptide is stable in the contact with blood. So before closing, I would like to just briefly say we made another really interesting and important finding recently in the paper, just got accepted. And we show, we ask the question, does spatial confinement of pro-inflammatory macrophages change their output, cytokine production? And we asked this question due to the following observation. If you start stimulating a macrophage with LPS, you see very soon that it gets round and it spreads significantly. And the spreading starts about 3 hours after LPS activation. What we can show now, if we grow macrophages on adhesive islands, that all the first NF-kappa B activation processes in response to LPS, they are all upregulated. But if you do not allow the macrophages to spread, and they can only spread by polymerizing the actin cytoskeleton, if you prohibit that by confining them, they greatly reduce the output of INOS, of IL-1 beta, IL-6, and other biomarkers that you often use. We also find that independently of your method of confinement, if you confine the macrophages in small pores, which do not allow them to spread, or if you confine them by cell crowding, again, they can't spread to this huge size. We see the same response. We worked out the mechanism, how this is working. MITFA binds to G-actin. Upon polymerization, it's released and it migrates into the nucleus. And we furthermore find that HDAC3 levels are less upregulated under confinement, and that the epigenetic profile signature of pro-inflammatory macrophages is affected by confinement. With that, I come to the last slide. Forces and physical factors co-regulate the transcriptomics and epigenetics of cells. This is not recognized in the community, and therefore you very rarely see which signaling pathways are regulated by these factors. And I hope with this animal study to have opened a door towards theranostic applications of this peptide probe. With that, I would like to thank a large number of collaborators, because so many technologies need to come together and so many different ways of thinking to make progress. And then I very much like to thank my research group who has done all the work, and you for your attention. Thank you.
Thank you very much, Viola, for a very interesting talk. I will allow a question. We're just about correct on time. There will be an additional 20 minutes at the end for questions and discussion, and Beat also has an important announcement. But so just before I put Beat on, are there any questions for Viola? Yes?
So thank you very much for a beautiful talk. I did want to ask you, do you think that the next generation of therapeutics will not only target the diseased cells, but also will target actually the ECM that is affecting the disease?
This is absolutely crucial to come, because imagine you create diseased matrix, you kill the cells that are residing in it, and we know already that if you reseed cells into a cancerous extracellular matrix, they turn cancerous very rapidly. So you leave this diseased matrix behind in every single one of the applications.